it was clear from the start that we wanted to bring in other types of data. So, for example, for some of this data, we have 3D structure data that we can model, and the idea was very much to jointly model both the amino acid sequences and the 3D structure, and many other kind of associated pieces of data. For example, the species that the protein was observed in. And I think this is really where, we all felt that BFNs had a chance to shine.
Welcome back to Let's Talk Research, the official InstaDeep podcast. I'm Massimo Bortone.
I'm Bora Gulola. Today we're continuing our deep dive into Bayesian Flow Networks, or BFNs, with Alex Graves.
Right, so in the last episode we talked about the philosophical and mathematical spark behind BFNs. If you haven't already, please make sure to check that episode, because today we are shifting gears.
That's right. Today we are discussing what happens when BFNs actually meet real-world data. We're looking at how we've used BFNs for protein sequence modeling, as well as antibody design, and everything in between. Welcome back, Alex.
Thank you. I'm glad to be back.
I want to ask you, when was the first time where you thought BFNs could really work with biological data?
So I started thinking about biological data quite early on in the process. I think partly because I saw it as something that would benefit from a non-autoregressive approach. And in some sense, a kind of a gateway for me was to start looking at what's called SMILES data. So SMILES is a kind of text format or a data format where molecular structures are written down as strings of characters, and they often contain characters like equal signs and brackets that look kind of like smileys, which is where the name comes from. And so, this was given that I'd already had a model trained on character level, text modeling, this was a very natural kind of segue into biological data. So yeah, even, before the paper was published, the first BFN paper, I started looking at, modeling SMILES data. And I think, it was already clear then that there was even though the data is written down as a left to right text string, it obviously corresponds to, something that occupies three-dimensional space in the real world. And for example, two characters that are very far apart in the SMILES string might be right next to each other in three-dimensional space. So it already seemed like a very natural place for a system that was non-autoregressive and potentially one that was kind of learning the order in which to model the data.
Great. I mean, obviously, that works really well, right? SMILES data looks a lot like strings of text. It is, in fact, strings of text, but it codes real kind of molecular structure, molecular connectivity. But we've also obviously done a lot of work on extending this into kind of more complex biological data sets like protein sequences with ProbBFN. Would you mind talking a little bit more about kind of how that emerged and what challenges were and how we ended up with the model, essentially?
Yeah, so I think, SMILES data for me was a sort of an easy starting point because, it looks a lot like text data. But, when I joined InstaDeep, there was already kind of a push underway to do a more kind of complete multimodal protein model. So, starting off with amino acid sequences, which again are essentially just text strings, so that part was, relatively straightforward. But then, it was clear from the start that we wanted to bring in other types of data. So for example, for some of this data, we have 3D structure data that we can model. And, the idea was very much to jointly model both the amino acid sequences and the 3D structure and, many other kind of associated pieces of data. For example, the species that the protein was observed in. And I think this is really where, we all felt that BFNs had a chance to shine, because they'd been designed from the ground up to be kind of, I want to say modality independent. That's maybe not quite true, but at least applicable to lots of different modalities, in particular to, modalities that are discrete or categorical, like amino acid sequences, and modalities that are continuous, like 3D structure and, like 3D points in Euclidean space, essentially. And having this single framework that could accommodate all of those different types of data just felt like a really natural way to start. Essentially what we were planning to do was take all of the data contained in large-scale protein databases and feed it all into a single model. So bring together, every piece of associated data about a single protein that could be found in UniProt or, similar databases, and, learn that whole thing with a single model.
Yeah, absolutely. And there's a lot of utility to having a model like that, but also just looking at the structure modality, so to speak, there's a certain advantage of not using an autoregressive model to model that, right?
Yes.
Because the sequence essentially stands for a structure, biologically speaking, where there are lots of long-range dependencies. And so if you're modeling it in an autoregressive manner, you're probably going to run into some challenges, right?
Yeah, I mean, for example, and obviously you're much more the expert on this than I am, but actual protein structures tend to curl up in space, and 2 points that are far apart in the sequence as it's written end up almost touching in reality. So it was clear from the start that the autoregressive, there was no real reason to favor an autoregressive approach here. It builds in a bias that isn't really present in the data at all. So I think everyone was in agreement that it was better not to look at this autoregressively. And then it was really a question of, well, what tools do we have? And, diffusion models were an obvious candidate. And the issue there has been, historically, they haven't done as well with discrete data. There are variants of diffusion models that work better. Lately, masked diffusion has become popular, although in my opinion, masked diffusion is more like autoregression in a random order than it is like diffusion. But these are questions of semantics, really. Essentially, there were many different approaches that we could have used to model this data, but it seemed that there wasn't a single framework that would accommodate all of these different types of data. And that was where we really felt that building this thing from the ground up around BFNs could be advantageous.
Yeah, exactly. I mean, the challenge really is to, as we said in the last episode, right, to have one single model where you can just chuck all the data in, just learns a rich joint distribution, and then you can do whatever you want with that distribution.
Exactly.
Right?
And I think it's really important to see that, when you start looking at real data like this, these distributions really matter. 3 Nobel Prizes last year were awarded for AlphaFold, which is essentially a model that of a specific conditional distribution, right? It's a distribution of structure given sequence. But there are, many other conditional distributions that you can get from the same data. You can do inverse folding, where you go from structure to sequence. You can do various kinds of classification and inpainting, and every single one of those actually has the potential to, be a scientific breakthrough, to really make revolutionary changes. So it's something that, this is where what to me makes this kind of research so exciting, is that these, quite abstract mathematical principles based around probability distributions, based around flow of information, based around joint and conditional sampling, those can have this real world impact, especially when you start looking at things like, scientific, biological data, health data, things like that.
Yeah, absolutely. And previously, when we've talked about this, you've kind of conceptualized this really nicely as a way to explore the data, right? These kind of joint BFNs are a really good way to essentially just learn from the data and then just be able to explore it.
Yeah, and so that's exactly, I think, the path that we've been following at InstaDeep. And yeah, I mentioned earlier, we kind of conceptualize this as a way to somehow empty the contents of a database and put it into a model. And I find this kind of fascinating that even works. That you can take this, for example, the UniProt database that's been collected by many different research teams gathering data in the lab, and then you can essentially distill all of that information into the parameters or the weights of a single neural network, given the right loss function. And once you have it, you kind of have, in a sense, a version of that database that you can query, you can ask questions of. You can ask it about probable things that aren't actually in the database but could be. You can say, well, if I took a protein that had this particular sequence or this fragment of a sequence that is present in reality, and then I asked you to generate the rest of the sequence, or I asked you to generate all or part of the structure or some of the tags or metadata associated with that sequence, it can give you possible answers. So I really want to stress this word possible.
Mm-hmm.
So I think a really key feature of this model is that it's entirely probabilistic. It won't answer a simple question with a simple answer. It won't say, well, if I specified this part of the sequence, tell me what the rest is, because there is no one answer, right? Mm-hmm. There's rather a distribution, a cloud of different possible answers. And the idea, if a model like this is well trained, what it will give you is many different possible candidates, all of which fit the constraints that you've given it. And it's really, this is where the kind of exploration part comes in. It's not an oracle that answers a question with a simple answer. It's rather something that gives you a feel for the set or the space of possible answers in some sense.
Yeah, absolutely. And one big use case one domain where we've found this to be really useful is antibody sequences, right? So antibodies are obviously, they have the sequence associated with them. So that's the heavy chain and the light chain. They come together and form the functional antibody. And within that, you also have these different regions of importance, right? You've got your CDR loops, which actually bind the pathogen or the antigen. Strictly speaking, you've got the framework which kind of hold the CDR loops in place. And then you've also got all this other data about the antibody, right? You've got the genetics of the antibody, the spliced together from 5 genes, each antibody. And there's a bit of random shuffling going on there as well, right? So it's kind of this combinatorial problem. Then you introduce mutations, so the sequence changes a little bit. But then there's also other properties that we're really interested in, especially in the clinic, right? We want an antibody to have nice biophysical properties, like it shouldn't be too hydrophobic, it shouldn't be too charged, and it should also be tolerable in the clinic, right? It shouldn't produce kind of a high risk of immunogenicity, and it should probably look like something that came from a human, right? Lots of different species obviously produce antibodies, and there's a lot of data out there. But when we're actually trying to develop a therapeutic, what we want is an antibody that binds its antigen. But beyond that, we also want to make sure that we can optimize for all these different things at the same time, right? We might want a certain genetic lineage, but at the same time, we might want well-behaved biophysical characteristics and so on and so forth. And that's where BFNs really kind of shine for us, right? We were able to put all of these things together, and then out comes this really nice model that we can essentially query.
Yeah, and that's definitely been, the most exciting part of the research for me over the last year, is seeing this, abstract model converted into something that's really functionally useful. And I think it is specifically useful because it's so open-ended. Like if you had to retrain the model, if you were still thinking in this sort of task-focused paradigm and had to retrain the model for each of these problems, you wouldn't get anywhere. It would take too long. It would be too cumbersome. And this idea that once you've got a single model trained once on the joint distribution, you can look at any there's this whole combinatorial space of things that you want to kind of fix and things that you want the model to generate. And I feel like it becomes this very interesting exercise in, what kind of constraints can the model satisfy, and as you say, there's so many, these come in so many different forms. I mean, some of them are kind of sequences of data. Some of them are, things like complex 3-dimensional structure. Other things are single tags, like you said, properties that you want a system to have. And, that's obviously an extremely useful area in all kinds of design. If you can say, well, generate me something that is good at X or has property Y, it's very easy to see a lot of practical use cases for that. And, these systems aren't magical. It won't be able to solve, if it's really impossible to create something with those properties, then the system won't be able to do that for you. But at the very least, you can explore everything that the model was able to glean from the data. And I think that in general, that's a lot more than we realize. Like I have a general feeling everyone is wowed by generative AI at the moment, but we've only actually scratched the surface of what it can do with real world data, with scientific data.
So, as you know, I'm not involved in BFNs at all. I'm more on the optimistic simulation modeling side of things at InstaDeep. But could you maybe, to make it a bit more concrete, describe how does this conditioning on these real world constraints work in practice? Like how do we actually achieve this from the model?
This is a good question. So conditional sampling isn't straightforward. The model, the original framework, the loss functions for Bayesian flow networks were set up to kind of optimize the joint probability of the data. You're basically kind of minimizing the negative log probability, standard kind of maximum likelihood loss function. And then the question is, once you've got that joint model, how can you fix certain variables and generate others? That's conditional sampling, essentially. And so most of the mathematics for this, which are actually fairly involved, but most of it can be ported over from the diffusion literature. And this is the advantage of Bayesian flow networks remaining in a continuous space, even when dealing with discrete data, because these techniques often involve looking at the gradient of the network with respect to, essentially, it comes down to you almost treat the data as a classifier of you treat the model as a classifier of the data that you want to condition on. So you kind of say, well, at each point during this generative process, the model can tell you, or at least the model can estimate the probability of the specific conditioning information that you're trying to condition on. And then you can use that information, you can backprop through the model, you can get a gradient, and you can use that to guide the generative process. And it's sort of weaves in a few different areas of kind of sampling theory, particle, for example.
Particle swarm optimization.
Particle filtering. Particle filters, are an area that people have used for similar kind of complex sequential data models. So it's mathematically fairly complex, but the nice thing is that most of this mathematical framework already exists. It's already been developed in other contexts, and it's just the case of kind of porting it over to the specific distributions that you get out of Bayesian flow networks.
Right. So essentially, the model provides kind of like an energy landscape.
Something like that.
If I'm trying to make analogies to the field that I'm comfortable with which is atomistic modeling. So I guess I'm kind of imagining it like as the model provides this energy landscape where you then, by conditioning, you end up in a valley. A respect to instead of another valley B, and then you maybe try to move around to explore a bit the landscape of your data. Is that?
Yeah. And you could think of it as there's a sort of energy landscape, and you want to guide this generative process towards the landscape that kind of satisfies the conditions.
Right.
So you want to you're pushing it at every step so as to make the conditioning information more probable. And that in the end is it means the model is forced to match the conditioning information. That's the easy part, of course. The hard part is to make everything else that it generates consistent with the conditioning information. And I find it quite fascinating the way it works. It's the same sort of iterative step-by-step generative process that you usually have, just with a kind of a guidance step. Every step is kind of guided in a slightly different direction by this conditioning signal. And it's very similar to, what people use, for example, to do, text-based image generation. Like they have a guidance signal from the caption, and they use that to direct the generation of the image.
And so let's say you question the model, you give the model some constraints, give me an output that fulfills these constraints. How would you then go about sort of measuring the confidence in the output that the model has given to you? Because I think that's often a problem that is still very much unresolved in generative AI, right?
Yeah, and I would say it's a problem that's much more kind of significant when you're looking at scientific data than it is at, for example, if your model, if you have a text, an image generation model, it's not really that critical to the user, what degree of confidence the model has in the image, as long as it's a nice looking image, basically. But in scientific data, it's not like that. We need well calibrated models that give us a sense of
much more risk involved.
There's risk. Yeah. You might have to make actual sort of real world decisions based on what the model says. And so the critical thing is it's always a probability distribution, like I said earlier. So even if you're doing conditional modeling and you fixed some specific variables,
right?
What you get back from the model is an ensemble of predictions over the other variables. There's always a distribution, and because there's always a distribution, you can measure the uncertainty, you can measure the entropy, you can say, if these variables are fixed, how confident am I that this variable will be X or Y, just by basically generating lots of samples and looking at the population that you get.
So in layman's term, you would say then if the model is very uncertain about a area that you're conditioning on, then you would expect the ensemble to be very varied.
Yes.
Right?
Yeah, you'll get many different looking samples with very different properties, and that's usually a sign. That's usually a sign that in some sense the model doesn't know what to do with the conditioning information. It hasn't been able to get a strong signal.
It just picks
and so
something
it's just kind of picking things randomly.
Right.
But then a nice side effect of that is the fact that you get distributions over your variables, right? Is that you can actually see if the model can essentially satisfy the constraint in multiple different ways, right? Sometimes you want multiple candidates for an optimization experiment to come out. So say, for example, going back to the antibody case, if you're optimizing for some sort of biophysical property, then it's actually at times really beneficial to have a model that doesn't always return the same answer over and over again, right? Rather than having just one solution, you have multiple solutions that you can then carry forward.
Yeah, and I think it's very interesting. Very often these models surprise you in a positive way. It may be that you thought, given some particular conditioning information, you expected it to give you a particular answer, and you find that it gives you a whole set of answers, and that many of them are not what you expected, but they're still valid. I saw this early on when I started looking at image in painting. I'd find, okay, I take an image, I crop a part of the image out, and then I ask the model essentially to reconstruct that using conditional sampling, and it would often reconstruct it with something quite different, but that also looked consistent with the region that I'd given to the model. And so it is clearly it's not the fault of the model that it didn't kind of match my expectations. It's rather the fault of my expectations.
Yeah, correct. I mean, we also found this to be the case with BFN2, right? We found that if we optimize for a specific property, say biophysics in a lot of these cases, we actually got answers that were very, very different from each other. So much so that we actually thought there was a problem with the model at some point. But then when we actually check those sequences and analyze them independently, we actually found that they were all behaving exactly as was hoped.
Yeah. And I think that's where this gets really exciting, is this open-endedness can allow you to query a model in ways that you didn't even think of when you gathered the data, you didn't think of when you trained the model. They occur to you later, and then you can actually discover something. Even though it's strange in some sense to say, well, the data was already gathered, the data was already in one place. You're not adding anything new. You're not going into the lab and sort of performing new experiments, but you're finding new connections in the existing data just by kind of querying the model and seeing what it does.
And you don't have to think about what you might be conditioning on before you start training the model, right?
I think that's very crucial.
That comes at a later step.
And I think from having talked to other people in the field, I think we've pushed this kind of conditional sampling boat at InstaDeep further than most. For a lot of people, when they talk about conditional models, they usually mean sort of one modality conditioned on another, like images conditioned on text or vice versa. And we've looked at these really fine-grained conditioning structures where, as you said, we're taking a specific region maybe this like complementarity determining region, and we're fixing that, and we're generating other regions of the same sequence. We found pretty much every permutation of conditioning information we could throw at it. There's a reason. There's a reason for that to be interesting, essentially.
Yeah. And so much so, right, that we've actually had to specify that there are so many different types of conditioning that you could use here, so many combinations, that we can't physically try them all out.
Yeah.
Right? We actually just need the community kind of play around with the model and see what comes out and what interesting things they can do with it.
I think this is a good example of another way of sort of decoupling two things, as I view it. So on one hand, you have researchers that develop and work on models. And then on the other side, you have people that apply these models in real world settings. And I think what this offers is a way of sort of maybe decouple these two sides a little bit. You don't have to, as a researcher, think so hard about the possible future applications. You can focus on your research, you can work on your model, and then offer to the people that work on applications actually a model that is very flexible and that people can actually play with, in a sense.
And I think this, mirrors what we've seen in the larger kind of generative AI space. People worked for years building these really powerful foundation models in language and in images and elsewhere. But it was only really when the tools like ChatGPT were made public and people started playing with them that they started finding all these uses. They started inventing these very clever forms of prompt engineering and ways to kind of manipulate the model and get it to do things. And so I think there is definitely this aspect of, almost like crowdsourcing or something that, you really want to build the most powerful and most flexible model you can out of the data, and then give people as much access to it as possible.
So obviously there's also this added dimension here of the fact that no two experimental setups are the same, right? Some labs will say, for example, going back to antibodies, they'll use libraries of candidates that are already pre-filtered for having developability issues. So that might be your biophysics and such. Other labs will just use a naive library, so to speak, right? A library that hasn't been filtered for these things. So having the flexibility of the model in terms of how we condition it means essentially that both of those labs can use the same model to optimize something like immunogenicity. But lab B, where antibodies haven't been pre-filtered, might just need to add this additional constraint during the optimization experiment. Hopefully the end result is still the same. So that's a really, really big strength of the model.
Yeah, and what we found is, adapting the model to a different set of data like that is very often the case of just kind of adding an extra field. You add this one extra field like immunogenicity and ask the model to kind of jointly model that along with everything else. And then you can kind of like toggle between these two settings. Like you can think of it as kind of like partitioning the data in some sense. And then you get this where people talk about kind of like shared statistical strength. Like the statistics of one of those data sets, they're going to be very similar to the other, just because one has been filtered or one is slightly different. You don't want to have to kind of run two separate experiments every time you slightly change the data preparation. You really want this one model that can, pick up the kind of shared signal across all of the data, and then moving between those different settings is usually just a case of changing the conditioning information. In a similar way, a language model can be trained on all different types of styles of writing. And then it can generate any one particular style just by specifying that in the prompt. You can tell it to write formally, you can tell it to write a poem, so forth. But having all of that language in a single model benefits every one of those kind of sub models individually. And I think you get the same effect here. Basically, you get this real synergy-like effect. Another thing, as soon as you move away from the kind of curated space of machine learning data sets where everything is perfect and all the images are cropped to the same dimension and whatever, there's a few problems you always run into with real world data, and one of them is missing data, right? You have data where there are fields missing where, like for example, or just, measurements were not made for more than a small subset of the data. For example, we have huge numbers of protein sequences and not that many, sort of verified 3D protein structures. And so it was a key sort of design constraint was to say, well, this system should be able to operate in the absence, with any part of the data missing, essentially. You can just treat it as kind of something that's masked out. And then even better than that, the model should be able to make predictions about anything that's missing. And I think this speaks to, for a lot of empirical scientists, missing data is really like a massive headache. People say, well, we've got these missing fields, but I don't want to throw away all of that data just because a few fields are missing in the middle. What do I do? Well, I'll do some kind of interpolation. I'll do something that fills in this missing data. I know it's not correct, but it means that then I can run my model and I can get a reasonable answer. But I think if you take this sort of holistic joint modeling approach, that immediately goes away. And actually what you could do instead is generate, a distribution of candidates for all of that missing data. But you don't even have to, right? The model will operate without the missing data. It'll just kind of like internally, internally it'll have a distribution over things that data could have been if it was present.
Yeah, and this is also something where we've seen movement in the field in that direction, where, for example, the next iteration of a model will be trained with a data set that is supplemented using predictions made by a previous iteration of the model. So it's really nice to just have something in place where that ability is already baked into the model.
Yeah. I think that's very important. And I definitely think being able to extend the models without fully retraining them is something that's going to become kind of the importance will grow as the size and the complexity of the data grows. At the moment already with sort of large language models, it's far too expensive to retrain them from scratch every time you have a new problem. So people have become very ingenious about how they take existing models and kind of wrap them with various forms of fine tuning. But I think that's something we can also take over into this kind of BFN framework for scientific data.
Yeah, and especially linking then models together also.
Yes.
I think this adds a whole other structure on top of models that are out there. So we've thought now extensively about what you can do with BFNs and their potential. Do you see them in the future going become increasingly important in the biological space?
I really hope so. Yeah, I mean, I think that it's been very exciting for me to work in that space. Actually, my father was a biologist, but interestingly, I never even studied biology at school. So the last year or so has been a learning experience for me in that sense. And I would very much like to see BFNs pushed further, especially in this space of kind of designing, candidate molecules, candidate proteins, and using really like exploiting this ability to fix these kind of sampling, these conditional samples. And just basically seeing where we can go with that.
Yeah, I mean, obviously I've had the joy of working with you on both PropBFN and AbBFN 1 and 2 now, which we've developed extensively, and we've also kind of made sure that they are usable for the community and out there. So if you want to play around with any of these models, feel free to find the links in our bio. We have code for PropBFN, code for AbBFN, weights are published, as well as a nice playground web app for AbBFN 2 that you can try out yourself.
Right. And so Alex, with that, we would like to thank you for joining us here and talk about your research. It's been really interesting to listen to your story and the success of BFNs.
Thank you very much.
All right, so in the next episode, we will be playing a bit more into my own strengths, and we will be talking about everything atomistic simulation and modeling with some of my fellow colleagues at InstaDeep. So make sure to pop by and see you then.